Okay, so any kind of difference in DNA letter sequence, which remember A, T's, C's, and G's, or amino acid sequence, remember there's 20 different amino acids. It all comes from mutations. So we have to have mutations to get any differences. Remember, we think of mutations as a bad thing, but mutations happen. Usually they are if you just randomly, which they are random, randomly change things. Normally things don't get better. But there are some times when they do help an organism survive. So, and clearly things have been surviving on Earth for a long time. And they occur gradually over time. And scientists have measured this very carefully, and they've developed a rate that we can expect mutations to occur. Say, one per day. Your DNA is always replicating, so there's always chances, small mistakes, small differences might accrue. And scientists have been pretty good at being able to nail down how many differences it take over a period of time, how many differences they expect to see. And that's what we call a molecular clock. And when we categorize organisms, the number of differences between groups is used to measure how long it's been since they split from a common ancestor. So for humans and something like the chimpanzee, that's about 5 million years before we were related, and then we branched off. Chimpanzees went one way, we went the other, and gradually started changing. We can calculate that distance, that time, by how many differences there are between our amino acids in DNA and chimpanzees' amino acids in DNA. And the type of DNA is preferred to use is mitochondrial DNA because it's very stable. It doesn't change as much as nuclear DNA, and it's easy to obtain. So most of these studies have been done using mitochondrial DNA. And the more differences we see means there's more time. Since the splitting, fewer differences, less time. So, for example, when we look at, you'll see in the phylogram about 70,000 years ago, humans of European and Japanese descent split off. So let me talk here. It's about 140,000 years ago. African and the European Japanese group split. So say, leaving Africa 140,000 years ago, this group came over here from Africa. Say this is Africa, and this is Asia, basically. And then they traveled around, and then boom, about 70,000 years ago, Japanese and then European split. So about 140,000 here, and then about 70,000 there. And between human chimpanzees, that's 5 million years ago. And here you can see in the little diagram on the phylogenetic tree of the humans right here. And we see chimps and then a different type of chimp and gorilla and orangutan and the different time period. So that's on 271, so you can write that down. Another couple terms, analogous and homologous. So homologous, they're similar because they share a common ancestor. And the one we talk about a lot is this pentadactyl limb right here. We have certain bones, and they're arranged in different shapes in humans, in bats, in dogs, in whales. If we look at those bones, all the same bones are there, just slightly different sizes, slightly different, or sometimes more different than others, but different sizes and shapes. But they're similar because of that common ancestor. And that's from this idea of divergent evolution. And what we normally think of in biology are the finches that Darwin made famous in the Galapagos Islands. A small group arrived in the Galapagos. That represents this dot. And then as they spread throughout the islands, they all changed to adapt to different conditions. So different species, they evolved over time to change and to utilize the resources. So that's what you see, it radiates out from the middle. Oh, I made a little smiley face. And that's from divergent. See, they're going apart. And this idea of analogous, they're similar even if they don't share that common ancestors. And that's something different called convergent. So a good way to think about this is the wing. You think, insects and birds, they're groups of organisms that have wings, and in a lot of ways, wings are very similar shape. That doesn't mean they share a common ancestor. They're very closely related. They just happen to, by mutation, natural selection, come up with the same sort of design to solve a problem, which is moving around their environment, obtaining food, maybe trying to escape predators, and they just happen to, over time, develop a very similar structure, but they're not closely related. So it can be tricky when we're looking at these things in nature. So we call these groups clades, and to make these cladograms, amino acid or DNA-based sequences, and the number of differences, the sequence is called parsimony, which is a fancy way of saying the more differences in DNA, the farther apart they're going to be put on a family tree. And a program calculates the most probable evolutionary tree based on the parsimony, which is the fewest number of differences. Branching off points are called nodes. That's N-O-D-E-S. And these nodes represent where we have a branching off into two different species. So here we have a nice cladogram, very nice, as you can see. Based on differences, actually I think this one's based on homology. I'm looking here. Yeah, so this is based on morphology. Sorry about that. So very similar, all these call them reptiles, but turtles, based on their morphology, are the most different. Then we got lizards and snakes branching off. So this is the common ancestor here, and it started going the turtle and non-turtle route, and then a group branched off to go the lizard, snake route, and then continuing. And then we have this really kind of dense one. We got birds, and then non-avian dinosaurs, and then crocodiles. And you'll see later on, Dinosauria would be this clade here, these birds and non-avian dinosaurs. And then archosaurs, we're going to include the crocodiles here. And then squamates, it's kind of hard to read in the handwriting or the color I chose here. TES, that's what this is, which includes the lizards and the snakes. So this is on 272. Give experience with how to make a phylogeny there. So they've been doing this for a long time, but it's only been, recently, relatively, molecular methods, and they've been doing a lot of changing. They've caused scientists to really rethink things when they get a better view of the similarities, because the morphology, which is the shape, can be deceiving, and they can look similar, but that could just be because of convergent evolution. So DNA is the best way to look at this, and the new ones really are improving, seeing evolutionary relationships. There.